Hello, ladies and gentlemen. Welcome back to Beard Biology. I'm Gaurav, and today I'm going to discuss about the glycolysis. Yes, the most important and the first reaction of respiration, that is glycolysis. So, without wasting our time, let me start. Glycolysis means glycose plus lysis. That means glycose means sugar and lysis means splitting. So glycolysis refers to the splitting of sugar. That means glycolysis means the splitting of sugar. The sugar is converted into the pyruvic acid. Now we will discuss about the place of the glycolysis, where glycolysis takes place. The place of glycolysis, that is cytoplasm of all living cells. Yes, glycolysis takes place in the cytoplasm of all living cells. The reactions of glycolysis, the steps of glycolysis, was first given by three scientists. Glycolysis was first given by three scientists, namely Emden, Meyerhof, and Parnes. So we just picking up the first letter of the name of the scientist, E M P. E M P. E for Emden, M for Meyerhof, and P for Parnes. The three scientists who discovered the glycolysis. So the glycolysis is also called as the EMP pathway. Glycolysis is the common path between aerobic and anaerobic respiration. Yes, glycolysis is the common path between aerobic and anaerobic respiration. That means we go through aerobic or anaerobic pathway, the glucose must be converted at first into the pyruvic acid. So glycolysis is required in both case aerobic and anaerobic respiration. Glycolysis is also called the EMP pathway. In the process of glycolysis, we see the glucose will be converted into two molecules of pyruvic acid. The glucose will undergo partial breakdown. Glucose will undergo partial breakdown or oxidation, through which it will convert it into two molecules of pyruvic acid. And in this process, this glucose is derived from sucrose or starch. Yes, this glucose which is used in the process of glycolysis is derived from sucrose or starch. Sucrose, which is the end product of photosynthesis, or starch. Now we will discuss about the steps of the glycolysis through which the glucose will be converted into the final end product of the pyruvic acid. At first, glucose will be converted. Glucose is now at first step. The first step of glycolysis, the glucose is converted into glucose 6-phosphate. Yes, the 6 carbon of glucose is phosphorylated in this step. This means in this process, one molecule of ATP will be converted into ADP. Mg2 plus ion is also required, and the enzyme which is required in this step is hexokinase enzyme. Second step, glucose 6-phosphate will now convert it to its another isomer, that is fructose 6-phosphate. Isomerase enzyme is required in this step. Isomerase enzyme is required. After that, fructose 6-phosphate will be converted into fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. This means the 6 carbon of fructose is phosphorylated in this step. It means one molecule of ATP is required in this step. One molecule of ATP is converted to the ADP, Mg2 plus ion is required, and the enzyme which is required, that is phosphofructokinase enzyme. Now this fructose 1,6-bisphosphate will be converted into two interchangeable isomers, that is 3-phosphoglyceraldehyde. It is a 3 carbon molecule, and dihydroxyacetone phosphate, that is DHAP. It is also a 3 carbon molecule, and these two compounds are interchangeable. After that, 3-phosphoglyceraldehyde, this means 2 molecule of 3-phosphoglyceraldehyde. It is because of DHAP will be converting to 3-phosphoglyceraldehyde. In this process, 2 NAD plus will be converted into 2 NADH plus 2 H plus. 2 molecule of water is required, and dehydrogenase is the enzyme which is required. 2 molecule of 3-phosphoglyceraldehyde will be converted into 1,3-bis phosphoglyceric acid. This means phosphoglyceraldehyde is converted into 1,3-bis phosphoglyceric acid. This means here oxidation is done. Removal of hydrogen, that means oxidation is done, dehydrogenase enzyme. Now 2 molecule of 1,3-bis phosphoglyceric acid will be converted with the help of 2 ADP converting into 2 ATP and the trans-phosphorylase enzyme 3-phosphoglyceric acid. 2 molecule of 1,3-bis phosphoglyceric acid converting to 3-phosphoglyceric acid. 2 molecule of 3-phosphoglyceric acid, and in this process trans-phosphorylase enzyme is required. Now 3-phosphoglyceric acid will be converted into 2-phosphoglyceric acid. 3-phosphoglyceric acid converted into 2-phosphoglyceric acid. 2 molecule of 3-phosphoglyceric acid is converted into 2-phosphoglyceric acid. 2 molecule of each. In this process mutase enzyme is required. After that, 2 molecule of phosphoglyceric acid will now be converted into 2-phosphoenol pyruvic acid. 2-phosphoenol pyruvic acid. In this process we see the enzyme enolase is required. The enzyme required is enolase, and in this step 1 molecule of water is removed. Now 2 molecule of 2-phosphoenol pyruvic acid will be converted into the end product of pyruvic acid, the end product of the glycolysis, that is the pyruvic acid. And this pyruvic acid will be participate in the next step of the respiration. In this process 2 ATP is gained. 2 ATP will be converted into 2 ATP. Pyruvic acid kinase enzyme is required in this process, and Mg2 plus and K plus ions is also required. Now in the whole process of the glycolysis, in the first half, that is from glucose to the fructose 1,6-bisphosphate, there is investment of 2 molecule of ATP. 2 molecule of ATP, and in the last half, that is from fructose 1,6-bisphosphate to the pyruvic acid, there is gain of 4 molecule of ATP and 2 NADH. So we can say the net gain is 4 minus 2, that is 2 ATP molecule and 2 NADH. 2 ATP and 2 NADH is the net gain of the glycolysis. The end product of the glycolysis, that is pyruvic acid, will now participate in the link reaction, where it will undergo through the oxidative decarboxylation and it will convert it into the acetyl-CoA. Acetyl-CoA will participate in the Krebs cycle. We will discuss about the Krebs cycle in the next video. Please do like, share and subscribe our video. Thank you.